So the objectives for the innate immunity module are listed here. This is the final of 5 mini lectures discussing this content. And today's lecture will be focused on failures of the innate immune system. So I'd first like to talk about complement deficiencies. And one of the things we noted about complement is that components of complement are great opsonins. It turns out that these opsonins are really important for encapsulated bacteria. And they are particularly important for clearance of a bacteria called Neisseria meningitidis. And so deficiencies with complement are most commonly associated with Neisseria meningitidis. If you see somebody with susceptibility to Neisseria meningitidis infection, you should be immediately thinking about complement. Now, there's other encapsulated bacteria such as Streptococcus and Haemophilus that are also more prevalent in people with complemented deficiencies. These are all encapsulated bacteria. So complement activation is particularly important for the clearance of encapsulated bacteria. There are also deficiencies associated in the alternative pathway with some of the inhibitors of complement. Remember, we talked about factor H and factor I. These are negative regulators of complement that destabilize C3 convertase at mammalian membranes. And of course, if you lose these, you can imagine that you might have overactivation of complement. It's actually interesting, though, because what ends up happening is that this leads to constant overconsumption of C3 to the point where you have very little circulating C3 left. And this overconsumption of C3 actually leads to an impairment of normal complement function. And therefore, people that have deficiencies in some of the regulators of complement, like factor H and factor I, are also susceptible to Neisseria meningitidis infection, just like people that have deficiencies in other components of the alternative complement pathway. Now, there are deficiencies associated with factor D and factor B, but they're incredibly rare. There's only been like a couple of families even documented with these deficiencies. Mutations in C3 are less rare, but also still pretty rare. But if you have mutations in C3, you're susceptible to a number of pyogenic bacterial infections. There are classical pathway deficiencies of complement. Again, these deficiencies are associated with susceptibility to Neisseria meningitidis. There are deficiencies within the mannose binding lectin pathway, such as MASP deficiency. These deficiencies are not nearly as severe. There may be some heightened susceptibility to bacterial infection during the first few months of life, but generally these lectin pathway deficiencies are rather modest. And lastly, there are deficiencies in the formation of membrane attack complexes, and this again leads to susceptibility to Neisseria meningitidis infection. So just reiterating the point, when you think of complement deficiencies, think of Neisseria meningitidis. There are some other key disease syndromes that are associated with complement deficiency that I'll talk about right now. One is called paroxysmal nocturnal hemoglobinuria, or PNH. So here we have an example of some of the inhibitors of complement. PNH is a complement-induced intravascular hemolytic anemia. And so you can get dark urine, especially in the mornings, and thrombosis. And it's a defect in a gene called PIG-A, or phosphatidylinositol glycan A. And this is needed to make GPI anchors that certain cell surface molecules use, and also is encoded on the X chromosome. It turns out that 2 of these inhibitory proteins, CD59 and DAF, decay accelerating factor, use GPI anchors to connect to the cell membrane. And so in the absence of this gene, you can't add these GPI anchors, and these 2 molecules are not expressed on the cell membrane. And this leads to overactivation of the membrane attack complex and periodic lysis of red blood cells. And one of the interesting things about PNH is you also have heightened susceptibility to Neisseria meningitidis, because constant overactivation of the complement system leads to overconsumption of C3 and therefore impairment of normal complement function. One of the really interesting things about this also, this is an acquired mutation. It's not an inherited mutation. A second disease related to complement is hereditary angioedema. This is caused by the loss, depression, or misfunctioning of the C1 inhibitor. And one of the points I want to make here is that whereas C1 inhibitor plays a key role in regulating complement, it also plays a key role in coagulation, the contact system, and the fibrinolysis system. And loss of C1H can lead to increased levels of bradykinin and periodic periods of rapid swelling of hands, feet, face, limbs due to this overproduction. It can become severe in late childhood. And so this is a picture of an individual undergoing an episode of angioedema. You can see the swollen cheeks and lips. In some places, the eyes can be swollen entirely shut because of this. There are some other key deficiencies of the innate immune system. We're going to talk about these three: chronic granulomatous disease, Chidiac-Higashi syndrome, and leukocyte adhesion deficiency. Chronic granulomatous disease is a redox defect. It's a defect in the function of the NADPH oxidase complex. The most common form of this is a defect in the GP91 subunit of this complex because it is X-linked. There are certain bacteria and pathogens that tend to live within phagolysosomes. Mycobacteria tuberculosis is famous for this. Bacteria that don't really get cleared by macrophages within their phagolysosome tend to induce the formation of these granuloma structures, which tend to be the formation, the general structure are large multinucleated macrophages called giant cells surrounded by T cells, mostly CD4 positive T cells. And these granulomatous structures are meant to kind of contain an infection that can't be cleared by the macrophage phagolysosome. Well, in chronic granulomatous disease, there's a whole variety of infections that now induce the formation of these granulomas because the phagolysosome is defective and is unable to clear these pathogens. And so these susceptibilities are actually quite severe. Chidiac-Higashi syndrome is another syndrome that results in defective function of the phagolysosome. This is an autosomal recessive mutation that results in recurrent bacterial infection with the same pattern of susceptibility as CGD. Also can result in loss of pigmentation. And the mutation in Chidiac-Higashi syndrome is in a gene called CHS1, or sometimes it's called the LYST gene. And it's a gene that's really important for the trafficking of granules and endosomal vesicles within cells. And so if you lack this gene, or this gene is dysfunctional, you have a failure to form functional phagolysosomes within the cell. These symptoms usually appear soon after birth, certainly in children under 5 years. A third key defect of the innate immune system is leukocyte adhesion deficiency. Leukocyte adhesion deficiency results from a loss of LFA1. LFA1, if you'll recall, is really important for trafficking of cells from the circulation into sites of inflammation. It's an integrin. And there's a whole family of integrins, and these different integrins are expressed on different cell types, but LFA1 is expressed on many, many cell types and is important for their trafficking. There's two subunits of LFA1, an alpha chain and a beta chain. Here we have an example of a patient with leukocyte adhesion deficiency. This is a flow cytometry plot. On the y-axis, we see expression of CD3, a T cell marker, and on the x-axis, we see expression of CD18, which is the beta chain of LFA1. And what you can see is this patient lacks any expression of LFA1 on its T cells compared to a control patient with no mutation. So this results in a failure of leukocytes to traffic to sites of inflammation, and of course this results in increased susceptibility to many types of infections. So those are some of the key immunodeficiencies associated with an innate immune system, but I want to turn your attention now to thinking about how viruses escape the innate immune system and suppress the innate immune system. And I decided to use it as an example responses that we've learned about in the past year to the SARS coronavirus 2, which is the cause of COVID-19. And these are in red, these little boxes in red are different viral gene products that inhibit various aspects of the response to this virus. And so here we have RIG-I. So remember, Rig-like receptors are cytoplasmic sensors, pattern recognition receptors that recognize viral double-stranded RNAs. And under normal circumstances, Rig-I will become activated and induce a signaling cascade that results in the recruitment of interferon response factors and even NF-kappa B, which results in the production of type 1 interferons like interferon beta. Interferon beta is then secreted, binds to a type 1 interferon receptor, and induces cytokine signaling within that cell that allows it to express many antiviral effectors that limit the spread of the virus. However, in the case of SARS coronavirus, there's a number of molecules that inhibit almost every step of this pathway. They inhibit the activation of Rig-I, they inhibit the activation of NF-kappa B, they inhibit the activation of interferon response factors, and they inhibit cytokine signaling induced by interferon beta in the target cell. Another way in which some pathogens can escape the innate immune system is through the induction of what's called a cytokine storm. And I'm again going to use SARS coronavirus as an example because it's been understood and appreciated over the last year that this coronavirus is very effective at inducing these cytokine storms. So under normal circumstances, here's a SARS coronavirus infecting epithelial cells. And these infected epithelial cells will induce activation of alveolar macrophages that detect viral patterns via their cytoplasmic sensors and start to make these cytokines. And these cytokines result in the recruitment of innate immune cells like monocytes and neutrophils and eosinophils. And these innate immune cells typically will enable the clearance of this pathogen or help with the clearance of the virus. In the case of coronavirus, they don't. And it's not really clear what the mechanisms are that prevent these cells from clearing the coronavirus in certain circumstances. It may be that if the innate response is somewhat delayed or suppressed, that by the time the innate response is recruited to the site of viral replication, it has already exceeded the ability of that innate response to control replication. It may be also that certain components of the coronavirus inhibit regulatory cytokines that suppress the immune response. And so what ends up happening is this response starts to simmer and then starts to spiral out of control until you get a huge production of cytokines that induce this effect that's called cytokine storm. One of the things that this results in is damage to surrounding tissue, including the accumulation of fibrin, weakening these blood vessel walls and allowing for fluid to seep into the lung cavity and leading to respiratory failure. Cytokine storms dysregulate the inflammatory response. So even though you have all these cytokines around, you're not able to make a normal innate immune response because these cytokines are unregulated or dysregulated. Cytokine storms can also occur during septic shock, for example. And lastly, there's a number of chronic inflammatory diseases that you will learn about throughout your first 2 years in medical school. These are often driven by a combination of environmental and genetic factors. They can be driven by overactivation of the innate and adaptive immune system. Inflammatory bowel disease is a classic example of chronic inflammation. Crohn's and ulcerative colitis are two flavors of that. Chronic inflammation can obviously have many deleterious side effects and has been associated with many diseases: lung disease, stroke, obesity, cancer, and even some neurodegenerative diseases. Chances are at some point in your career you're going to have to learn more about how inflammation affects diseases that you are treating because it is so widespread and is such a critical and important component of disease pathology in so many systems. So these are the objectives from our innate immunity lecture. You're going to want to review these objectives and make sure that you have acquired the material that you need from this innate immunity module.